Namaskar dear students, welcome you to our channel by Gulab Sir. As a part of series of lectures, today's topic is cell division, especially mitosis. We already learned about interphase. Now we have to learn about mitosis. बहुत इंटरेस्टिंग पार्ट है जो आपको हमेशा एक्साइटमेंट में पढ़ने के लिए मन करता होगा कि सेल आखिर कैसे डिवाइड होते हैं, नए सेल कैसे प्रोड्यूस होते हैं, और थोड़ा सा कंफ्यूजिंग भी रहता है अगर अच्छी तरह से क्लियर नहीं हुआ तो। सेल डिवीजन के 4 प्रकार है, अमाइटोसिस, एंडोमाइटोसिस, माइटोसिस एंड मियोसिस। उसमें से आज अपन माइटोसिस पढ़ेंगे। What is mitosis? It is a equational division. That means when two new daughter cells are produced, they have equal number of chromosomes. That is what I have shown here. Suppose take the example, they have 2 sets of chromosomes. We divide and produce 2 cells has equal number of chromosomes, that is 2 sets. You can see here the same way I have shown here. That means a cell division in which when 2 new daughter cells are produced which has similar number of chromosomes to their parental cell is known as mitosis, and hence it is called equational division. How it happens? When our suppose cell which undergo mitosis and produce 2 new cells, from here the 2 sets of extra chromosome comes, and that is very important क्योंकि हमारे अंदर 46 क्रोमोसोम है। फिर 92 होते कैसे ताकि हर सेल में 46 आए, वो सीखना बहुत जरूरी है, वो समझना बहुत जरूरी है। देखिए यहाँ पे ये मोनोहेलन क्रोमोसोम है। मोनोहेलन मतलब हैविंग वन आर्म ऑन आइदर साइड of centromere. So here you can see one arm on either side of that. Now during interphase, that we learn in preparatory phase, interphase, how cell get ready to divide and produce new cells. That is what production of protein, RNA and DNA material double to have the 2 cells. जैसे कि घर में 2 भाई का बंटवारा होता है तो दोनों भाई को 2 घर, 2 चूल्हे देना होता है, सब कुछ देना होता है। वैसे 2 सेल बनेंगे तो उसमें 2 सेट्स ऑफ क्रोमोसोम तो चाहिए, साथ साथ में सभी ऑर्गेनल चाहिए, साइटोप्लाज्म चाहिए। वो कब होता है? प्रिपरेटरी फेज में। यहाँ थोड़ा सा समराइज किया गया है ताकि हमको डिटेल में बाद में पढ़ना है। क्रोमोसोम नंबर 92 होते कैसे? क्योंकि हर एक सेल में 46 चाहिए, तो यहाँ देखिए 46 है, तो यहाँ जो सिंगल मोनोहेलन क्रोमोसोम है वो बाइवेलेंट बनता है। दैट मींस during G1 phase and G2 phase protein and RNA are synthesized, and during S phase of interphase DNA replication occur, and hence that DNA become double, and hence that monovalent chromosome become bivalent. You can see here having 1 arm on either side, here you can see 2 arms on either side. These are the 2 sister chromatids. Say 23 chromosome from mother, 23 chromosome from father, and that is 1 chromosome is equal to 23 when we take the scale. These are 46 chromosomes. So these 46 chromosomes become double by their chromatin material, chromosome material. Later these bivalent chromosomes, what I have shown, get split into 2. See, these chromosomes get split into 2. These get split into 2, and thereby each represent the new cell. And you can see here after splitting of that one, these cell receive one of these 2 and one of these 2. And same way this cell receive one of these 2 and one of these 2. Hence these 2 cells are similar to parent. See here, this cell is similar to parent, this cell is similar to parent. So this is what, that nuclear material get double and each cell receive equal number of chromosomes, say 46 as parental cell. Hence it is called equational division. Now we have to understand in detail. Before that, मुझे ऐसा विश्वास है आप हमारे वीडियोस बहुत पसंद आ रहे हैं, आपको आप देख भी रहे हो, लेकिन साथ साथ में लाइक और सब्सक्राइब किया करिए तो मजा आ जाएगी, और भी अच्छे वीडियो बनाएंगे। वही हमारे लिए इंस्पिरेशन होता है, मोटिवेशन होता है, ताकि हम अच्छे से अच्छे प्रिपेयर करके अच्छे से अच्छे इजी वे, कितने इजी वे आप पढ़ सकते हो, समझ सकते हैं, उसकी तैयारी करके आपको पढ़ाएं। वही एक ये प्रयास है। शायद आप जान भी गए होंगे कि मैंने डायग्राम कैसे निकाली और आपको कैसे समझा रहा हूं, तो आप समझ सकते हैं। तो पुनः एक बार कंटिन्यू करते हैं। लेट अस कम ऑन नेक्स्ट पेज। As I told you that mitosis occurs in 2 steps which we have to learn in detail. First step is karyokinesis, second is cytokinesis. You are aware about karyokinesis, that is division of nucleus, cytokinesis, that is division of cytoplasm. Karyo matlab nucleus, kinesis means division. So here karyokinesis occur in 4 substages that are prophase, metaphase, anaphase and telophase. That is what we have to understand in detail. Question can be asked, what is mitosis? Explain these 4 phases in detail with diagram and 2 2 to 3 characteristics about each. That will be 5-mark question. Even in NEET, very intelligent questions are asked. If you understood concept, I am sure if you see this video, 100% if any question asked on mitosis, you will solve it. Now let's go. Our next page. See, this is the diagram what I have drawn. I said you कि आप इंटरेस्ट तो ले रहे लेकिन लाइक और सब्सक्राइब नहीं कर रहे. अगर वो भी करेंगे देखो कैसे अच्छे हमको मोटिवेशन मिलता है, हम कैसे रिप्रेजेंट करने के लिए प्रयास करते हैं. कितना इजी है देखो. सी दिस फर्स्ट स्टेज इज प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज सीक्वेंस में लगाए गए हैं. अभी आप समझ सकते हैं कि माइटोसिस है क्या. सी इन दिस स्टेज आफ्टर इंटरफेज chromosome material already become double. Therefore, I have shown 2 chromatid with each chromosome on either side of the centromere. You can see, you can visualize here. Now you guess here, other organelles are also present there. Nuclear membrane is also intact, and that is what cell of interphase enter into the prophase. So in the early stage of prophase, all they are there, but continuous. When prophase continue, at the end of prophase, nuclear membrane get disappear, nucleolus get disappear. Say this is a nucleolus, which is I have not drawn, but now to show. Here there is a nucleolus. This nucleolus get disappear. Nuclear membrane get disappear, organelles get disappear, and spindle fiber formation begins. Moreover, prophase, on the onset of prophase, with the continuous process, condensation, coiling and shortening of chromosome, so that these chromatin fibers become shortened like this, so that they can be easily split. If they are long enough, they will entangle and will not split enough, will not move to the opposite pole. Therefore, they need to be shortened. So during mitosis process, chromosomes are condensed, coiled and shortened and become highly thick in the end of the prophase itself, and then cell enter into the metaphase. Now see difference between these two, prophase and metaphase, for which already I have given in detail about that characteristics. You go through this as a notes, that will be helpful to you, which I am explaining here with this diagram is written there in notes. So don't worry about notes, notes are given there, but see here, see the difference. These two cell you just compare and you will find here organelles are there, organelles are not there. Nuclear membrane is around chromosome, nuclear membrane is disappear. Nucleolus disappear. So what is left? Only cytoplasm and chromosome gather in the equator. So metaphase is the second phase of mitosis, in which nuclear membrane, nucleolus and organelles are disappear. Centrioles move towards opposite pole, that begins in prophase itself, and they reach to the opposite pole so as to form the spindle fiber, and spindle fibers are formed over here. These are the spindle fibers. These are the spindle fibers, and even this aster formation you can observe in animals. So during metaphase, spindle fibers are completely formed. Chromosomes gather in the equator. All 46 chromosomes, they gather in the equator in a one plate, in a same plane, and therefore it is called as metaphasial plate or equatorial plate, and that is mentioned in points. So this is for the gathering of this chromosome in one plane at the center is called metaphasic plate. At the same time, centromeres, that centromeres attach with the spindle fiber, where there are centromeres having 2 plates called kinetochores. So centromere covered by kinetochore which attach to the spindle fiber in metaphase stage. Now this is what metaphase stage. It is ready to enter into anaphase. What does it mean? At the onset of anaphase, spindle fiber begin to contract. As soon as they begin to contract, chromosomes get split into 2, and hence they begin to move towards the pole. These kinetochores which were present over the centromere, they get joined, attached with spindle fiber, and thereby when spindle fiber contract, this bivalent chromosome gets split into 2 monovalent, this bivalent gets split into 2 monovalent, hence that 46 become 92, and thereby that each cell can receive 46 chromosome that I did explain to you. अभी आपको समझ में तो आया होगा कि 92 क्रोमोसोम हुए कैसे, 46 क्रोमोसोम के 92 क्रोमोसोम हुए कैसे, ताकि each cell has to get 46 chromosome in this way. So metaphase, anaphase. At the metaphase stage, chromosomes are at its highest stage, highest thickness, and therefore they can easily appear under microscope. Moreover, morphology of the chromosome is best studied in anaphase stage, because with the contraction of spindle fiber, when kinetochores move towards the opposite pole. They drag, they pull this chromosome towards opposite pole. Hence, they look either V shape, L shape, J shape or I shape. Thereby, the morphology of the chromosome is best studied in anaphase stage. Please remember this for NEET. Thickness of the chromosome is at the highest in which stage? Metaphase stage. In which stage the morphology of the chromosome is studied? Anaphase stage. In which stage that chromosome moves towards opposite pole and becomes 92? Anaphase stage. That is what you have to remember. So splitting of chromosome occur at the onset of anaphase, and thereby bivalent chromosome become monovalent, and thereby 46 chromosome with 2 chromatid each become 92 with 1 chromatid each, and that is what the ratio. So 1:2, here 1:1. Why I am saying this 1:2 and 1:1? Please remember, ratio of the chromosome to chromatid is 1:2. Here ratio of the chromosome to chromatid is 1:1, and that is very important for NEET. Question can be asked. What is the ratio of chromosome in metaphase to the chromatid? Is 1:2. Each chromosome has 2 chromatid. Here each chromosome has 1 chromatid on either side. That is also very clear to you. Now let us know about telophase. Is the last phase of the mitosis. Karyokinesis is completed. Chromosome reach to the opposite pole. Spindle fibers are disappeared. Chromosome begin to uncoil, unelongated, so as to be as it is in the prophase stage. Interphase stage, nuclear membrane reappear, nucleolus reappear, all organelles are reappear. That means that one cell, parental cell, divided into 2 cells with equal apparatus. That cellular apparatus which required for that cell to survive. Both the cell has equal number of mitochondria, approximately equal number of mitochondria, Golgi body, endoplasmic reticulum, lysosome, all organelles get equally divided. No doubt, after karyokinesis, cytokinesis begins. That is what we have to learn in next. And same this diagram you can observe on net. See there, these 2 chromosomes. Here new daughter cells has produced with same number of chromosomes. Therefore, it is equational division. As I did explain you, same it is shown over here. Please go through and study it. See now, I have been given this point as I have discussed with you. Same points are written. Go through. Each is very easy language. Moreover, I have made it very clear how monovalent chromosome become bivalent during interphase, and later this bivalent chromosome they condense and coil at the onset of metaphase. During prophase, condensation and coiling begins, and thereby they become bivalent. Therefore, I have written under the heading of prophase that monovalent become bivalent, condense, coil and highly thicken, that at the end of the prophase. So here these are the points written. You go through. I just made it to have the fast reading. This is the first phase of mitosis. Beginning of the prophase starts with the condensation and coiling of chromosome, and that is shown here. You understood? As a result, chromosome become very thick and they are visible and bivalent. In that stage, centrioles start moving towards opposite pole. Centrioles means what? Let us understand why this diagram is very important. What are centriole? Centriole present just very close to the nucleus. This is one bundle of tubulin protein. This is another bundle of tubulin protein. These are the 2 centrioles. They are perpendicular to each other and covered by amorphous clear cross centrosome. So these 2 centrioles are present in centrosome. They begin to move to the opposite pole to form this spindle fiber, and that is what you please understand. See, next, that centriole split during prophase and move to opposite pole to form the spindle fibers. At the end of the prophase, nuclear membrane, nucleolus, and other organelles are completely disappear, and thus we enter into the metaphase. See here metaphase, that is the second phase of division. Nuclear membrane, nucleolus all are disappear, spindle fibers are formed, and aster formation, that is what star shape, appear at the polar in animal cell. Because spindle centrioles are present in animal cell, absent in plant cell, so aster formation appear in only animal cell. That is also a point of NEET. Next, all chromosomes gather at the plane, and that is called equatorial plate or metaphasic plate, that I did explain you. Spindle fiber attached to the kinetochore which cover over the centromere so as to when contraction occur, they get split to have the two monovalent chromosome from bivalent chromosome. At metaphase, chromosomes are at their highest thickness and hence they are observed under microscope, as well as their morphology can be studied at the advances. Anaphase stage is the third stage of the mitosis. Spindle fibers begin to contract and thereby, see, I have shown bivalent become monovalent, bivalent become monovalent with the splitting of the centromere, that kinetochore, each carrying one chromatid with that chromosome. So bivalent turn into monovalent. As the contraction of the spindle fiber begin, chromosome get dragged, pulled towards the opposite pole, and thereby that bivalent gets split to monovalent. At the same time, when kinetochore attached with centromere move towards the pole, that means chromosomes are dragged and hence their morphology can be studied. Either they are V shape, L shape, J shape or I shape. For your knowledge, V shape are metacentric, L shape are submetacentric, J shape are acrocentric, and I shape are telocentric. Thus, the morphology of the chromosome is best studied in anaphase, and that is very clear to you. So finally, chromosome reach to the opposite pole in telophase, and in telophase, nuclear membrane reappear, nucleolus reappear, centriole set at the site of nucleolus, spindle fiber disappear, all organelles reappear, cell plate formation begins, and that is what constriction in the cytoplasm called cytokinesis. Hence, next stage is the cytokinesis. What is cytokinesis? Karyokinesis is followed by cytokinesis, that is division of the cytoplasm of the cell into two equal halves to represent cellular apparatus along with nucleus, and hence two new daughter cells are produced with equal amount of cytoplasm and all other organelles, and thereby two daughter cells are in functional stage, and thus two new daughter cells are produced from a single parental cell. This way that mitosis is completed. So please you go through all these notes which I have written in the points and come to the conclusion that mitosis is nothing but a equational division, where parental cell undergo division and produce two new cells with equal number of chromosomes. I hope you did understood it and enjoyed it. Again and again you can see it so as to get it very clear. As notes are given, that notes are sufficient to write in examination. If question asks what is mitosis, explain it for 5 marks. I hope you can understand. Definitely in next video we are going to learn about meiosis, so as to you will get clear both mitosis and meiosis, and in third video we will have comparison between these two to have the best understanding really what is mitosis and what is meiosis, where mitosis do occur, where meiosis occur, and what are their significance. So it is a part of series of lecture, and in a sequence we shall learn. Thank you. Happy learning.